Bioinformatics.
Okay, and welcome to the lecture on short read alignment / mapping. So before I get into what exactly read mapping is in the context of next-gen sequencing, let's take a second and just revisit next-gen sequencing methods in general, so we kind of have some concepts that will be important for how mapping works and why it's valuable. So you've heard me mention paired and unpaired reads throughout the labs. And so what does it mean when you read a paper and it says something like 1 by 125 base pairs or 2 by 150 base pairs? And so that 1 and 2 times refers to whether it's unpaired, in the case of 1 they're single reads, versus paired where they have some type of mate, some set distance away. And so here's an example of kind of what those look like that we've shown you before, where you've got a read 1 and a read 2 that are sequencing opposite ends of the same fragment, and they end a set distance apart, so that when you think about how these reads would fit onto a genome that they were sequenced from, there you would have some information at the beginning and the end, as well as a fixed distance between them. How do you get there? Well, if we think back to library prep for many sequencing methods, you start with a set of whole DNA or RNA, and you randomly shear it into pieces. And then you take those pieces and potentially do a size selection where you only keep pieces that are a fixed size so that you know the entire fragment length. And then you sequence a fixed distance from either end so that you know exactly how many unknowns should be in the middle. And so that's how you end up with this kind of nice paired setup. And why is this useful? So there are lots of regions of genomes where there are repeats or high regions of similarity. And so in instances where you have, for example, this repeat here where you've got this green then blue and then this green then blue here, if your read spans a green and a blue region, it could potentially map or fit onto either one of these two spots. So that's kind of the dilemma of using single reads. If, however, you have paired reads and they are long enough, they can be valuable for kind of bridging those gaps into regions of non-similarity, so you can have some external information to figure out where it should fit. And so to illustrate that, here's that same example but with a paired read. So now one side is in this green-blue region and one side is in this blue-red, and now we know unambiguously that these two pairs should fit here. There's no way for it to fit over here. So now we've actually provided more information and we have more certainty in where our reads map or align to in the reference genome. And so really the goal of sequence alignment or read mapping is to find a home for all of these little short fragments that you sequenced onto a reference genome or something similar. Now, this can be complicated by a number of things. Repeats are one thing, and we'll talk about those. But there's all sorts of different variation in genomes. You could think about structural variation as far as deletions, insertions, the movement of mobile elements. You could also have duplications, whether they're tandem, whether they're shifted around. You could have rearrangements like translocations and inversions. All of these things can make read mapping and alignment both more challenging but also more informative. The other thing that you can kind of get out of read mapping is sequencing a bunch of individuals, for example, mapping them back to a reference and looking at which individuals differ at which sites from that reference. And so you could start to look at SNP or SNV variation. And for those of you that don't know what a SNP or a SNV is, a SNP or a SNV stand for single nucleotide polymorphism or variation. And so they're pretty much the same thing. For whatever reason, the community has decided that we call it a SNP, a single nucleotide polymorphism, if it's present in more than 1% of the population. Otherwise, it's called a variant or variation. I don't make the rules. The methods that we learned earlier for kind of pairwise sequence alignment or even multi-sequence alignment are great when you're dealing with small-scale things. They do a fantastic job for aligning relative to your scoring matrix. However, if you're talking about millions or billions of little tiny fragments trying to find a home on a huge genome, they just don't scale. And so we need different methods for that. And so why focus on this part of read mapping versus alignment? Well, I think it's important to understand because virtually almost all downstream analyses will, in addition to having quality control at the beginning of receiving your sequence data, will probably have some sort of read mapping or alignment step. Oftentimes the workflow for genomics involve sequencing your data, getting your FASTQ data, quality controlling that, and then aligning it to a genome or a transcriptome or some other reference. And then from there you can do all sorts of things. Find out what types of SNPs and SNVs are in your populations of interest. If it's RNA, that's when you would quantify transcription throughput. You could find peaks related to methylation patterns. All of these things rely on accurate mapping. Okay, so say you've got your cleaned up FASTQ file. What do you do next? Well, read mapping is really just essentially finding a home for every read in that file, if there is a home. And then this is important because you need to get that alignment just right. Otherwise, all your downstream analyses don't really mean anything because they're built upon false information. And so this is challenging. We've already talked about that genomes vary widely in size. They can be huge. It's not unusual to have a genome that is over 3 billion base pairs long. Your sequence data can, from a single run, be over 100 billion reads or fragments sequenced. And so multiply that by the read length. If you're talking about 2 by 150 base pairs, that'd be 300 times 1 billion. That's 300 billion pieces of information. And on top of that, genomes can be exceedingly complex. So this is a challenge. One extra challenging piece of information are repeats or repetitive regions. Over half of the human genome, or roughly half of the human genome, are transposable elements. And so these are things that either use a copy-paste or a cut-paste mechanism and are littered throughout the genome. And so any reads that map to just within a transposable element are going to map to multiple places within your genome as well. And so that's another challenge. And so you could almost think about genomes as being kind of this puzzle with lots of reflections and regions of similarity. And so a good analogy might be thinking about reads as being a puzzle that you're trying to put back together. Where do each of these pieces fit? But I like to think of it as being far more complex than that. It's really as if you have multiple images of the same thing that have reflections within themselves that are all stacked together, and maybe you're missing pieces of one and you've got extra pieces of another and they're strewn all about, and then you shatter it into 100 billion pieces and try and fix it. And so that's really what the challenge of read mapping and alignment is at a genomic level. And so when we think about these complicated situations with large amounts of data, these slow pairwise traditional alignment strategies won't work. Remember, it took several seconds to do some of our single pairwise alignments. If you're doing 3 billion bases, you don't have a lifetime to wait. And so these things are always being optimized, but you could think about a pretty decent rate for aligning huge numbers of things being feasible, doing 10,000 per second, something along that, maybe 20 or 30,000. And that kind of should boggle your mind a little bit, that you're able to take all these pieces, tens or 20s of thousands of them, and find their best home using an algorithm per second. So something that comes up is, what is the difference between mapping and alignment? And really the short answer these days is that there isn't a huge difference, because most modern read mappers or aligners tend to use a little bit of both kind of strategies. The longer answer is that aligners are basically a higher throughput version of the traditional alignment we talked about. They cut corners, but they still try to find the best base pair to base pair alignment, typically kind of like a local style alignment. While mappers take a totally different approach and use what's called a hash table to try and find the best spot in the genome for pieces of reads to find where they're supposed to go. Almost similar to what we saw with BLAST. And so I like to show kind of how things work. Again, I don't expect you to memorize these things, but it's nice to know how things work. And so hash mapping is really built upon indexing a reference first. And so part of today's lab is you will index things several times. So I want you to understand what's happening when you index something. So if you've got a very small genome, let's say a 16 base pair genome, to index it, we go back to our old friend, the sliding window. And we set our window size equal to k. And so oftentimes people will talk about the size of things as k-mers. In this case, a k of 3 we would call a 3-mer. If it was 5, it could be a 5-mer. And so if we wanted to hash out based on 3-mers, we could do a 3 base pair sliding window. And what happens in a hash table is not only do you get all those k-mers, you also get their genome positions that they start at. So cat starts at position 1, ATG starts at position 2, and so forth. But what's interesting about a hash table is when you get back to instances of the same k-mer, you just tack on an extra position here. So cat is at position 1 and 7. Okay, and we could go through TGG is at position 3 and 4, GGT is at 4 and 11, etc. And so this is a hash table. And so when you index your reference sequence, when you're getting ready to do read mapping, this is what is happening under the hood. Now, how this works for mapping is once you have your indexed hash table, every single read is quickly broken down into k-mers. And so here we've got our first 3-mer, TGG. All we have to do is go, where is this in the hash table? It is right here, and it could fit position 3 or 10. And so we go, okay, hash match, 3 or 10. What's our next 3-mer? It doesn't even have to be a sliding window. We could just jump to the next 3. This one, TCA, position 6. Well, there is only one possibility where this should fit because it's a multiple of 3, so we would expect 3 to follow by 6. Okay? And so this is the only place where we would expect this read to map. This didn't involve any alignment, any computation beyond comparing this against the hash table. Okay, and so that's a really simple example that doesn't take into account sequencing error, quality of your sequence, as well as actual biological variation. And so if we had a mismatch here, we can also kind of scale this as well. And so what do we do with this when we get to one where there is the possibility of fitting 2 different spots? Well, we could, rather than think about a scoring matrix and all sorts of complicated things, we could break it down to a simple edit distance as a way of evaluating things. How many changes do you need to make it fit something? And so here, an edit distance that has 1 substitution, curled to hurled, is an edit distance of 1. If it's a deletion or an insertion, and there's just one of them, it's an edit distance of 1. So we keep it very, very simple for efficiency's sake. And so that TCT is an edit distance of 1 away from being TCA or TCG. And so that kind of information is used to make decisions about if there's 2 possible places to fit something, the one that has the fewest edit distances will be the one that it will be placed at. So in this example, I used a k equals 3. But what's the right k to use? Should you use k equals 10, k equals 1? K equals 1, well, there's 4 possibilities, right? It's A, T, G, and C. K equals 3, there's 4 to the 3rd possibilities. If you go to k equals 10, you've got over 1 million possibilities, and each one of those is present in the human genome at least once. So it takes a really long k-mer to find things that will be unique, that will only map to 1 possible place. It does ramp up pretty quickly, so there is kind of this exponential growth in that. But the long answer, or really the short answer is, this is a very young field. Genomics, bioinformatics has only been around for about 15, 20 years in kind of its current state. And people are still figuring out what are best practices for some of these things. When is a K of 3 appropriate? When is a K of 90 appropriate? What's better for assembly? What's better for read mapping and alignment? So oftentimes you have to play with these things and hope that you will stumble onto really kind of the best algorithm for your specific complication as far as it relates to your mapping problem. One of the things that we need to do is we also need to evaluate how good our mappings are. So if you're allowing edit distances, you don't want to just throw reads places and then forget that they weren't exact matches. Or you don't want to forget that these things are short, and the shorter something is as far as a k-mer is, the more likely it could map to multiple places. And so there is going to be a mapping quality score associated with every read and where it goes, our confidence in where that read could go. Now, this is nice. They use a map Q score for this, and I just show you this because it shows that everything we learn isn't just specific to that thing. This is actually very similar to the Fred scale. They just changed the front name. It's still a one letter code that corresponds to -10 times the log 10 of your p-value for being mapped to that location. And so kind of same cutoffs. We want a map Q score of 30 or greater typically to consider that read as being mapped. Again, repeats. These are the biggest, really the biggest issue. They are everywhere in the human genome. They will suck in reads because reads that have repeat regions within them are going to map to so many different places. This leads to reads that have repeat sequences as a part of them having lower map quality score because they are capable of being mapped to multiple locations. And another problem is the quality of your reads themselves are involved in where things can map. So if you start with reads that have a quality score of 20, you already have lower certainty in any given base. And so that will be reflected in the certainty of that read being mapped to places, because typically you're going to map your FASTQ files, and for a good reason, the quality of individual base calls are also added into this. And so if you start with garbage sequence, you're going to get a garbage mapping set. And so we're going to explore in lab two different kind of popular read mappers or aligners. One is BWA and one is Bowtie2. I like to mention BWA, one, because it's really widely used and very, very popular, but also because it's kind of a fun example of some of the shortcomings of peer review. This was authored by Lee, and Lee attempted to publish this years ago and got really crappy reviews. They were almost kind of hostile. And when it was rejected, he was frustrated and just decided not to publish it and made it publicly available. And so, well, it's one of the really highly cited pieces of bioinformatic tools with 2,400, and I think this is very undercited. Almost everybody uses this. It was never actually been peer reviewed. And so I think it highlights some of the shortcomings of peer review in general, that things can be very high impact without going through peer review or even not making it through peer review, because it's the opinion of 2 or 3, maybe 4 people. So when you map things, this is what it would look like visually. And so at the end of this lab, I'll show you how to take some of your outputs from your read mapping and visualize them. And this is an application called Tablet. There's also integrated genome viewer. But you can see this is our, up at the top, this is basically our reference sequence. And then all of these are our reads. And here I've clicked on one, and you can kind of see the map Q score of it. We'll talk about what the cigar score is as well. It's a paired read, so we can also see its other mate, as well as the insert size between them, which is 442 base pairs. And if you look up here, you can see the window I'm showing you is just a small window of all the reads aligned to this very small genome. This is a very small example data set. But just look at these thousands of little tiny fragments that are actually all, hundreds of base pairs. And so this is what it looks like visually. Now, I want to show you what the actual data look like. And so the data you get from read mapping or aligning is typically in a SAM or a BAM format. This is a text-based way of representing both the sequence mapping or alignment, as well as lots of other pieces of value information that this here actually pulls from. How does this image or visualization know which other read is the pair, what the insert is between them, what the quality score is, all these things. This is all contained within the SAM and the BAM format. And you can also query this, as I'll show you in a little bit. Okay, and this is what the SAM format looks like. And so we've got every entry is basically one line. It's wrapped around here. And so I've designated the start of each line with green here. And so this is all one big SAM line. And you're going to have one of these for every single read in your alignment or read mapping. And so this looks like garbage. And so what you should ask yourself is, why do we want such a weird, crazy format? And I think the better thing to ask is, what would be a really great way to use tons of disk space and a very inefficient analysis? Well, the answer is to have a text file for every single alignment of each read to the genome. You would have 100 billion different alignments. And so that's not feasible. So we need to think and use kind of a creative way of containing all that information as compressed as possible while still being human readable. And so SAM and BAM formats look funky at first, but they're actually great. They're compressed. They are easy to manipulate. They can accommodate single and paired in data. They're simple to make, and there's plenty of great tools for working with them as well. And so I'm going to walk you through a little bit about what goes into a SAM format. You don't have to memorize this. I don't just sit down and read these things, but understanding the format and what types of things you can do with them is kind of the theme of this class, is that just knowing what's capable to be done out there will help you if you're ever thinking about, oh man, what can I do for this? Or how can I do this? Oh, I remember SAM format might have some of that information. Let me start reading up on how to pull it out. And so the SAM format is actually a column format. It is a table. It doesn't look like it because it's long and it's wrapped around in your terminal, but it's a tab delimited table. So this first thing has a tab before the second, before the third, all the way through, and it can be of varying lengths, different numbers of columns depending on what you want to do. But here's an example. The first column is the name of the read or the pair. And so this is the read name. The second column is the flag score. We'll talk about that. The third column is the reference sequence name. If you're thinking about a eukaryote, often this will be probably a chromosome 1, something like that if it's fully assembled. And then the position on that chromosome, as well as mapping quality. And then what's very helpful is the cigar string. And so there's other information. I'm not going to go through it all. But as an example, if you, for some reason, wanted to pull out the insert size and think, okay, based on my mapping, what's the average distance between my different pairs? Did it actually match the insert size that I know from doing the library prep? How would you go and pull out from a SAM file all of the insert sizes from all your 100 million reads and average it, for example, or look at the standard deviation to get a good sense of whether things are mapping as you would expect? Well, you've actually already done something like this, because if you remember, we had a table. It was comma delimited when you looked at it, but it very easily could be tab delimited as well. And this is the same layout, the same structure. If you wanted to pull out just the 9th column, that is the calculated insert size, you could use a regular expression on your SAM file and pull that out and pipe it over to a histogram generation script, something like that. It would be very easy to do if you wanted to spend a little bit of time just looking at some example code of how to make graphics. And so this is the power of these file formats and regular expressions. So I also want to mention what the second column is, because this is hugely informative. The second column is weird. It's called the flag column, and it has its own table of meaning. And it's basically a bunch of true-false. So the first one, the read originated from a paired sequencing molecule. In other words, was it part of paired sequencing? If it's true, you would put a 1 in the first position. If it's false, you would put a 0. You would do this for all of these different rows. Example, is the query the first read in the pair? Is it the forward read, in other words? That would be true or false. If it was the reverse read, it would be false. And so you end up with a number like this. And so this is weird, but each one of these you replace with 2 to that power, starting at 0. And you add them together. And so this will give you a unique number that can't be repeated except the same combination of any number of true and false of these. So here we've got 2 to the 0 plus 2 to the 1 plus 2 to the 5th plus 2 to the 6th. That's 64 plus 32 plus 2 plus 1, which is 99. Our flag score is 99. There is no other way to get 99 except this combination of components. And so this is a ton of information all contained within just a couple digits. The other really nice compressed piece of information, again, because we're not having whole alignments contained for every single read, is the cigar string. And so the cigar column basically tells us what differences there are between the read and the reference starting at its start location. And so what it is a breakdown of all the matches and mismatches. And so here we've got, from the reference versus the actual read that's mapped, we've got 4 that match, and that's designated as 4M. Then we have 1 deletion relative to the reference, so 1D. Then we have 2 more matches. 2M, we've got 2 insertions, 2I. Then we've got 7 matches and 2 substitutions. Now in this case, a match just means that there's not a deletion or an insertion in a really basic cigar string. It's not designating specific mismatches. There's an extended cigar string for that as well, but we'll keep it simple for now. So the alternative to the SAM format is the BAM format. It is more compressed, but if you ever head it or cat it, it's not readable. It's in funky letters and shapes and stuff. SAM is the human readable, if you believe that, version of the BAM format. Why do I bring this up? Because some programs want you to give them a BAM file, some want you to give them a SAM file. At the end of the day, it doesn't really matter because it is very, very easy to convert between them. This is how easy it is, once you understand the principles involved, to make a SAM file and then convert it to a BAM file. We need to run something like BWA and make our hash map of our reference genome. Here we've got human chromosome 38. So we're going BWA index this chromosome. And then from here, we could add some different options. But we could say BWA, use your MEM algorithm. -T, I believe, is number of processors to use, because sometimes these things can take a lot of compute power. With this reference, map this read and this read to it. And save that as a sample.sam file. And then if your next analysis says, no, I want a BAM file, well, you could just use something like samtools view and say the argument big S little b is saying I have a SAM file, but I want to convert it to a BAM file. And you just redirect the output to a BAM file. This seems maybe like foreign language right now, but it's once you've done this a few times, this becomes very easy to just repurpose and use. And this is really, everything I talked about is done right here. And then you could take this BAM file or the SAM file, visualize it, or put it into downstream analyses.